It is my great pleasure to introduce a friend and colleague for today's seminar. So coming to us from University of Florida is Ramon Alain Miranda Quintana, who's here giving a talk on a variety of things. So he has a background in radiochemistry, but also got his PhD in chemistry. He's from Cuba, so he did his studies in Havana. So with those two topics, it's amazing that we actually got you over to the States with those two things on the forums. But he's a rising star in the field. True story. And that I heard yesterday. But one of the things that's really spectacular about me, about Ramon is he was recruited into University of Florida, which has a long history of having really strong theoretical underpinnings rooted in the quantum theory project. And so when they had the opportunity to recruit Ramon, he came in and it was right during the beginning of the pandemic. And so getting his lab started was wrought with challenges, but with a very, very strong mathematical background, he was not only able to continue great momentum in his fundamental quantum theory projects, but also be able to branch out and really break new ground in a lot of areas that have people talking all the way from biology to drug discovery. And so some of the things he's going to talk about today, I'm particularly excited about simply because it offers new mechanisms to think about how we process and relate to the immense amount of data that's generated, for example, through MD simulations. And so I thought this was a particularly opportune time for him to present to this audience, because as the field moves forward, I think we need to think about not only how we manage experimental data, but how we also are able to integrate new types of theory data into the same common infrastructure in order to be able to move science forward. So let me also talk about how great Ramon is in terms of awards and things, since I'm sort of embellishing things, but let me just start out. I'll mainly get to some of the point. He's a rising star in the field, and he's received a number of awards. At University of Florida, he won the Oak Ridge Ralph E. Powell Junior Faculty Enhancement Award, and recently the OpenEye Cadence Molecular Science Outstanding Junior Faculty Award in Computational Chemistry. He's been recipient of a NIH R35 grant, and has a huge portfolio of very diverse topics, from fundamental methods developments to broad areas of collaboration involving the transfer of the methods that he's developed into different contexts. So without further ado, I'll let you kick it off and tell us about what you've been working on most recently. So thank you.
Thank you so much. There is absolutely no way that I can live up to our introduction, so it's essentially going downhill from this point onward. I do apologize. There are a few anime references sprinkled through the presentation, so I hope that you enjoy those. So, is it okay? Like okay. Yeah. So, as Darren mentioned, my group has been working on several topics. So I was hired to do quantum chemistry. Then I betrayed the people that hired me and started doing molecular dynamics and drug design. The title of the presentation is heavily inspired by work that Darren did like very early in his career. So for people that do electronic structure, you know that these methods are very, very computationally demanding and they are very computationally expensive. A way to scale that to bigger, to larger systems, essentially to literally break down your system into different like individual pieces and then trying to combine that at the end. That's the very popular and extremely powerful divide and conquer strategy. In my lab, when we deal with comparisons with similarity with protein structure and drug design, we take the totally opposite route. We gather everything and then we try to conquer the problem. And I try to like make the argument that that is also a very nice strategy. Like how do you gain a lot of like efficiency, time, and memory wise? So first of all, let me talk about things that I'm not going to be talking about today. So these are the other two type of projects that we have in the group. This is more connected to what I did during my PhD. It's essentially studying how charge and spin transfer can dictate chemistry in solution. We have been applying that to studying different specific ion effects on different protic and aprotic solvents. We propose this like a strong weak acid-base principle, essentially to explain things like the Hofmeister series for ions. We have applied this to essentially trying to discover new small molecules that are essentially stabilize and destabilize tertiary structure of proteins and biomolecules in solution. We also, as I mentioned, I was hired to do quantum chemistry, like actually like trying to solve the Schrodinger equation for complicated systems, essentially for strongly correlated systems. More particularly relevant for the audience, one of the systems that we are looking into are ferredoxins. Essentially, everything that either you're breaking and forming a lot of chemical bonds at the same time, or system that you have a bunch of like unpaired electrons that essentially spread over different nuclei. Our method, our recipe, our trick is the flexible ansatz for n-body configuration interaction. We obviously try extremely hard to spell fancy with the acronym. Fear not, there are going to be plenty of amazing acronyms along the talk. We are mostly focusing on combining coupled cluster wave functions with quasi-particles, essentially geminal wave functions, so we can describe these systems. These are the part of the group working on that: Patricia, Rugwed, Christina, Carlos, a postdoc, and we have a package to do this, which we call FunPig. So in 2020, in the extremely amazing year of 2020, I joined the faculty at UF in February of 2020. It's like a couple of weeks before the apocalypse. And then I was supposed to be working on this. However, I was alone in the group for a full year. I was trying to find ways to keep myself busy. So then at some point, I decided to change to something different and going very away from essentially from quantum, from charge transfer and whatnot, and start working essentially on what I think is just data science. Now, data science, which we have found a lot of different applications in chemistry. So when you think about similarity in chemistry, I like to think that basically this is something that people even doing alchemy, like from alchemy times, were like keeping in mind. Essentially given to this, like, I want to say one of the fundamental dogmas in chemistry. If you have similar molecules, you expect them to behave in similar ways. So for instance, if you have suffered through organic chemistry, and we have all suffered through organic chemistry, you know, you can tell a lot of things about the molecule in the middle because it's similar to the one on the right and it's similar to the one on the left. At a more fundamental level, perhaps the best example of similarity in chemistry is nothing more than the periodic table, which is essentially just an arrangement of the building blocks of nature based on how similar they are, how they share common properties. Okay? The way that I'm going to be talking about similarity is how do we calculate similarity between atoms, like small molecules, and like bigger biomolecules using computers. For that, we need to be able to represent molecules and entities in a way that we can actually process them using computers. How do we do that? Well, first of all, I'm going to be using a very general representation for molecular structures. So essentially, to me, a molecule is nothing more than a vector that has different components. Now, depending on the domain of application, there are going to be different ways in which we can represent different molecules using different strategies. For example, for drug-like molecules, for small molecules, a very prevalent strategy is just using vectors, which the components are just going to be either a 1 or a 0, either like an on bit or an off bit, which in the language of people doing drug design, these are called molecular fingerprints. There are several different recipes to generate molecular fingerprints. Let it be the Max Kiss, the extended connectivity fingerprint, or the Morgan fingerprint. The RD kit comes with like their own like preset set of fingerprints. Now, when you're thinking of something bigger like a protein or like DNA strand or like something like RNA protein complex, you are usually thinking about vectors in which the components are instead real numbers. And in these cases, use like standard ways of representing a structure include just like the standard Cartesian coordinates or internal coordinates. We can apply also this representation to small molecules if you use like some sort of like latent space representation, or even like in the QSAR community, just like some like molecular descriptors. Now, essentially everything that I'm going to be talking about today applies to both. So it's not limited only to the binary representation, it's not limited only to the continuous representation. I am going to be showing you examples of applications of the underlying framework to both ways of representing molecular structures, essentially ways of representing chemical information. With this, we can do things as like even like trying to refine protein structures, which is like extremely like relevant to the audience, but also like how to navigate, how to map, how to get chemical insight from what is known as chemical space. There is one intermediate case that I'm really not going to be talking about a lot today, but that we have also shown that we can apply these same tools to those cases. So in between having two characters, or in between having like infinitely like continuum many characters, there are other ways of representing chemical information, essentially using a finite alphabet, which is extremely popular if you're studying peptide protein sequences or DNA, RNA. So in one case, you have essentially 20 characters to build your representation. And in the other one, you have essentially just like 4 or 5 characters, depending if you're doing like DNA, RNA to study your systems. We have shown once again that these tools also apply to this case. We have like one paper showing how to use this like studying and dissecting like different type of like genetic relations between different families of proteins. Once again, I'm not going to be focusing on the part in the middle. It's mainly going to be like small molecules or like bigger molecules. But just keep in mind, if you're studying sequences, these tools are also like applicable to your problems. Okay? So with that in mind, also like the tools are so general that we even can go beyond just like your normal chemical structures. We have been collaborating with like this other group at UF, showing that you can use this type of like ways of dissecting data to study a mass spec experiments. So they have like this like imaging mass spectrometry technique. They generate hundreds of gigabytes of data at every single experiment. So this will give them, this actually gives them like a very like nice way to essentially to sort through all of that information, essentially finding the most relevant, like and physically meaningful like compounds like in that in those sets. So molecular comparisons, how do we compare molecules? I'm going to be showing first the case of binary input, which is the simplest one to explain and to understand. However, it's easily like transferable to other cases. So if we have two molecules, both represented with binary fingerprints, you essentially only have to calculate 4 different things. How many times you have 2 ones in common, like the common number of on bits between the 2 strings, the common number of off bits between the 2 strings, and obviously the mismatches, like the 0, 1, 1, 0 occurrences. So with this extremely simple like set of ingredients, it is pretty clear that A and D are going to quantify the quote unquote similarity between your 2 molecules, and that B and C are going to quantify the dissimilarity between your 2 molecules. Essentially, now it all boils down to mixing these 4 ingredients in a way that gives you a number that the bigger the number, the more similar the molecules. Essentially, we are looking at functions that are monotonically increasing with A and D and monotonically decreasing with B and C. There are obviously infinitely many choices for this. The drug design community have essentially railed behind the Tanimoto index, which is the one in the middle, which is nothing more than A divided by A plus B plus C. But there are other possibilities as well. So the so-called mission index right there on the left, the Baroni-Urbani booster. So once again, like essentially any function that increases with A and D and decreases with B and C is like a possible way of quantifying similarity. For the purposes of just appealing to the drug design community, I'm mostly going to focus on Tanimoto, but once again, everything that I'm going to be saying is also applicable to other ways of quantifying similarity. At this point, I want to stop for a second and focus on a totally computer science. I have no idea what's going on. Thank you. So at this point, I want to focus on a total computer science problem that is like how difficult is a problem. So if you are a PhD student, you think that your PhD is by far the hardest problem ever that humanity has faced. And that is true. But actually, we can quantify how difficult is your PhD. Actually, we can quantify how difficult is every possible problem. But from a mathematical / computer science idea, we can see how many resources we will need as a function of the size of the input of your problem. And so let me give you an extremely important problem. So Frodo had to destroy 1 ring. And that was arguably a really difficult problem. Now, you can argue that Frodo will have essentially the same amount of effort if he had to destroy 6 rings instead. Meaning that the amount of effort that Frodo had to put into his like amazing task is essentially independent of the number of rings that he had to carry to Mordor. Okay? And if you haven't seen the movies, you should definitely see the movies. Okay? Now, let's talk about walking. Your input size is the distance that you want to walk. And your resource is the time that it takes you to walk. So if it takes you 1 second to walk 1 meter, you expect that it will take you 2 seconds to walk 2 meters. Essentially, this is a problem in which the amount of effort that you need to put is proportional to the size of your input. Now, if you want to paint something, let's say 1 square of 1 by 1, and you need 1 bucket of paint, if you want to paint a surface of 2 by 2, you need 4 buckets of paint. So essentially, in this case, the amount of resources is proportional to the square of the input size. So in very precise mathematical terms, painting your house more complicated than destroying the One Ring, depending that your house is big enough. Okay? So this is what in computer science people call as the big O notation, that essentially allows you to classify the difficulty of a problem. We have problems that go like from very, very complicated, like factorial or exponential complexity, to essentially trivial problems that are O1. Basically, you want the exponent, that function of n, to be as small as possible. And this is going to be critical for the rest of the talk because I'm going to be comparing different algorithms to solve essentially the same task, and they differ in the computational complexity. And we will see in practice how big of a difference can make to go from something like O(n) squared to O(n). Okay? So with that, like a YouTube intermission of like bigger complexity, I'm trying to sell you on Tolkien. Let's talk about like how to quantify chemical diversity. So I showed you before how do you do comparisons between 2 molecules. And that is an incredibly important problem. However, in many cases, you have more than 2 molecules. So if you are doing generative design, for people that do generative AI for drug design, this is what they will call the internal diversity problem. So you have a library of molecules, sometimes with like millions or billions of molecules, and you want to see how they are related to each other. The standard approach to do this essentially boils down to finding every single pair of molecules, calculating the similarity between every single pair of molecules, and then like finding the average of that. Okay? If you have a set of conformations, let's say for a protein or like DNA, RNA, this is essentially the same problem, now rephrased as the conformational diversity problem. If you have 2 of these structures, like people in the community seem to love RMSD for some reason, it still is like it's totally unclear to me. So if you have many different conformations at the same point, you essentially need to find the average of all the possible pairs of RMSDs. Now, no matter if you're doing small molecules or bigger molecules, the literal first step is finding every single pair of compounds that is n choose 2, that is a problem that scales as n squared. So no matter how you're quantifying your similarity, if you're doing it just by using pairs of molecules, that is an intrinsically O(n) squared problem. How does this translate to life and time? The Campbell library, at some point, this is like 3 years ago, they had like roughly like 1.9 million molecules. They curate an extremely efficient software. This is like C++, parallelize all the nice things. If you want to calculate this average separation within every single molecule in Campbell, that will take you more than half a day, using once again pairwise comparisons. Now, my group was looking at this problem, and we came out. This is in 2021, so things were already getting better for everyone. So we proposed what we know as the extended similarity framework. I'm briefly just going to like speed run through this because now we have a better framework. But I just want to like paint you a picture of essentially the key ideas like underpinning this whole framework. So essentially, you will have n number of molecules, which you can arrange in a matrix. First step is always going to be adding every single column independently. So you will generate this vector, this sigma vector, and essentially every single k sub something is just the sum of all the elements in a column. So essentially, every single k is just counting the number of ones in that column. At that point, we propose to calculate a descriptor that is delta. Then the user had to define this parameter gamma, which we call a coincidence threshold. You compare this delta with gamma. If delta is bigger, then we say that that column is like contributing to a similarity, if not to a dissimilarity of the different molecules. You need to find a weight, like partial coincidence of 1 and zeros. For the similarity case, that's the f of s. For the dissimilarity, that is the f of d. And then you assign these to essentially representations of A, D, and B plus C, and you have your potential solution. Once again, like we are really not doing this right now, mainly for one reason. So the red points, the red indices that you see on the screen, those need to be user-provided parameters or functions. So the user had a lot of saying in how this was going to be operating. For the case of macromolecules, the recipe was even simpler. Just remove the square root from RMSD. So if you remove the square root from RMSD and now you want to combine any number of molecules, you just need to store the matrix of coordinates, the matrix with the square of the coordinates. You add all the columns independently, and then like some like kindergarten level algebra gives you a formula that essentially only depends on the sum over all the different columns. In both cases, the critical step is just adding all the columns. You are adding n columns. The problem scales linearly with the number of molecules in your set or number of conformations in your set. So once again, like we wanted to overcome the difficulties, essentially like we wanted to remove the user from the equation for the standard similarity. So then recently, oh yeah, and with this, you get like essentially like a global like way of quantifying diversity in your set, which you can use like to study like different linking criterion to like pathway analysis and so on and so on. Okay. So with this in mind, we came out with an instant similarity or ISIM. I will get back on the name later. So basically, Kenneth and David, like former postdoc in the group, we realized that we can get exactly the same number as the pairwise average. Just once again, oh, sorry, we will just add once again all the elements. But now the key realization is that if I know the number of ones in every single column, K choose 2 is the coincidence of like every possible coincidence of ones in one of these instances. If I know the number of ones, I know the number of zeros, that is n minus KQ. And once again, like n minus KQ choose 2 will give me the coincidence of all the possible zeros. And if I know the number of ones and the number of zeros, then I know the number of mismatches. And this is it. Like the user has absolutely zero saying in this, because this will like immediately tell you the possibilities for the 1, 1, 0, 0, and 1, 0 coincidences. Now, we take these expressions, we go to the formula to any similarity index like Tanimoto. Whenever you have A, you have coincidence of ones. Whenever you have B and C, you have the mismatches. You plug this into your formula, and you got yourself an OM method for essentially for quantifying chemical diversity. This is the comparisons on in the x-axis. You have the ON squared numbers. On the y-axis, you have our numbers. Each single plot has 10,000 different libraries. You see that you essentially get literally the same number from the ON squared formalism to a linear formalism. What is the obvious like critical application? The first application, once again, the problem of quantifying chemical diversity. Now, if you have a set of molecules, any number of molecules, you can calculate this number in one step. What is the practical like difference with respect to what we saw before? Well, I showed you before 1.9 million molecules, extremely optimized software, takes you half a day to calculate. We sent our like really like primitive Python code to the people at Campbell, and they were like the people of Campbell, they were like kind enough to test our approach, and it took them less than 4 seconds to get the same number. So you do see a huge speed up, essentially in quantifying diversity of these like bigger sets of molecules. So essentially, we have this like first tool that tells you how many things are connected to each other. Can we get also some local information? And yes, I mean, we can leverage this like global tool to get some local information, realizing that if I have a set of molecules, and if I remove one and calculate the similarity of what is left, I can have an idea of the importance of that molecule in the whole set. I can repeat this for every single molecule, and we get what we know as the complementary similarity. And essentially, if we rank these complementary similarity values, we can have an idea of which molecules are central to your set and which molecules are essentially outliers in your set. So the lower complementary similarity means you remove something, the average similarity decrease. So that is a key molecule for your set. You remove something from the set, similarity like essentially doesn't change. That is an outlier. So once again, an OM method to identify high density and low density regions in your data. We also, once again, we can apply this to protein conformations. Basically, the same principle. You will be like essentially seeing the effect of removing every single conformation from your ensemble, and you rank based on the complementary MSD values. Once again, what are the more important conformations? What are the outlier conformations in your set? The second application that we look into it's a critical problem, like a quintessential problem in drug design that is like, if I have, sorry, if I have a huge set of molecules, how do I find a diverse set of compounds from that set, from that like bigger set? We propose an extremely simple algorithm that essentially boils down to like pick the molecule that minimizes the Ising value. So we compare this with like the tools that are implemented, say in RDKit, the maximum algorithm. You see like in like orangey, like yellow, are the similarity values that you obtain with RDKit. In blue, the ones that you obtain with our method, you will see that consistently, like our method is finding more diverse subsets than the ones that you will find with RDKit. So not only a faster method, it also gives you like more insight. It gives you like a new way of finding like more diverse like subsets, like subsamples in your data. We also apply this once again, like to the case of macromolecules. We study like several systems. We ran like several tests from the green line to your right. Those are using tools that are currently available in the Amber package in CPP Trash. So essentially, if you were going to do this with Amber, you would like do clustering, and then you essentially find the representative inside every cluster. What we found is that if we quantify the RMSD as like the average RMSD as like a metric of separation between the selected molecules, we not only find more diverse subsets in general, but it only takes like around like 3 hours to process all these systems. While if you use hierarchical clustering, that will take like more than 200 hours to complete the same task. So once again, like you do see that speed up from the ability of like essentially comparing everything and selecting stuff all at once. In the case of chemical space exploration, we have applied this essentially to explore. These are different epigenetic focus libraries. We have applied this to essentially to showing how you can find the more important compounds in these like very large libraries. We have leveraged this into different ways to do sampling in chemical space. Like if you want to sample from the center of your library to the exterior, if you want to sample only focusing on the outliers, if you want to do mixing, essentially like a huge variety and flexibility of ways to do subsampling in a set, all of them linear scaling, and especially tailored to your potential applications. In the particular case, and this might be like particularly relevant to the current audience, in the particular case of protein folding, a critical problem for people running MD simulation is that once you run your MD simulation, how do you find your potential candidate to correspond to a native structure of that simulation, of that particular protein or protein like peptide complex or protein RNA complex? We propose this pipeline, Prime, that essentially also is going to leverage this idea of like sampling to identify outliers and high density regions. So first of all, you normalize your coordinates to make your life slightly simpler. And your first step, you can choose to remove the noise from your data. And we define noise at like some percentage of the outliers of the low density conformations in your set. You pick one of the similarity indices that I have been showing. Once again, you have like multiple ways of doing this. And then essentially, the critical idea is that you find your best representative not only by looking at the center of your clusters, you find your best representative by essentially finding in your main cluster which is the conformation that is furthest away from all of your other clusters. So once again, it's slightly more elaborated than just, okay, so I cluster my conformations, I find the highest populated cluster, and I find the center of that. Okay? So we benchmark different ways of essentially finding these most representative conformations. We have 6 possibilities. Like number 1, just find the centroid of everything. Like don't even bother doing clustering. Step number 2, which is what people usually do nowadays, so you will do some sort of clustering. You find the most populated center, the most populated cluster, and just like take the centroid of that. And also, as I mentioned, like different ways of quantifying the separation between molecules in the bigger cluster and the remaining ones. So you're essentially using information about all of your data and not only your main cluster. So when we try this over multiple systems, so we tried this for low temperature replicate change, high temperature replicate change simulations for like more flexible proteins, and for protein peptide systems, like the bigger the blue bar is, how are we like outperforming the traditional method. So you can see that essentially for every single of the systems, we are outperforming. We're finding structures in the like inside those MD simulations that are closer to the experimental structures than the ones that you will find if you just take the average of everything inside that main cluster. Okay, so basically could be seen as a way to do like a structure refinement. Like it's not exactly refinement, but like finding more more representative conformations within that like full MD simulation. Okay. So an obvious application of all of this is essentially clustering. So since it's the very first paper that we submitted on this strategy, like the very first referee said, like, hey, like why don't you just like apply it? You're talking about similarity and you're talking about groups of things. Why don't you just like apply this to clustering? So the obvious idea, at least like to us at the beginning, the very obvious idea to do clustering is like if at some point I have sets of molecules or sets of conformations, I can try to see which ones to combine if I try to maximize the similarity of the union of the two sets. So this is essentially a hierarchical agglomerative, like a bottom-up clustering strategy. We apply this once again to several systems. These are two test cases, a model protein DNA interaction system and like a model protein G system. What you have on the y-axis is the V measure, that is essentially a metric that quantifies how good is going to be your clustering. So the bigger V value, the better your clustering is going to be. You see that our method on the far right, we are essentially same quality, if not outperforming the traditional approaches, once again, by just being like faster in getting to what solution. An advantage of our method, since we are directly quantifying the separation between the different sets, we can literally propose a metric that quantifies the distance or the tax that you need to pay to join two different sets. This is like delta S metric. And if you monitor the values of that delta S, you can have potentially an idea of when to stop your clustering. So you start seeing like if you are combining structures like at the very early stages, you are really not paying like a lot of effort to doing those combinations. However, when you get to a point in which you are combining two sets that really should not be together, you see a huge jump in this metric. So the method itself can give you some idea of when to stop that clustering. To me, the most appealing result in this like first paper on clustering for ND simulations is the full analysis of the folding landscape of this model protein. So this was like a relatively simple yet non-trivial case. So this protein is known to fold following two different pathways. When we threw this problem at Amber and to CPP Trash, like CPP Trash like failed to find anything different than like totally random conformations and the totally folded state. However, when we apply our methods to the system, we were not only able to find the totally unfolded and the totally folded states, we're also able to identify all the more important metastable states along both protein folding pathways. So essentially, this was the paper that convinced me, first of all, to start asking money for NIH. Basically, what essentially like motivated me to start like doing like more and more MD, like with these tools, and not only drug design. With that, we have been like full on implementing like a trillion clustering algorithms. We are putting together a package that is already like available, like MDense. I did promise you amazing acronyms. This is yet another one of those. So in MDense, Lexing, which is an absolutely like amazing student, she put together a lot of functionality. So you can not only use our tools, but you can use them in a most flexible possible way. So we have all the pre-processing and the post-processing that you might need to interface MDense with your favorite software package. This is also included already, in MD analysis. So if you are used to using MD analysis for your simulations, you can use MDense exactly as you will use MD analysis. It can also interface with CPP Trash. So once again, it's like you run your Amber simulation, you run your GROMAX, you run your sham, whatever you want. You can jump, convert your files to whatever we're using, and then use all our methods. Prime, this like a structure refinement method is already included in MDense. However, the core of the module are essentially these like different clustering algorithms. We have hierarchical clustering, as I showed you before. We have radial clustering. We have like a density base, essentially like a reimagination of DBSCAN using our tools. But the one that I want to focus on is our version of K-means, not only because yet again it's a killer acronym, it's K-means nanny, but also because it allows you to essentially like see the power of like doing multiple comparisons at once. So if you are using normal k-means, which are usually k-means is sold as like a very efficient way of doing clustering. However, exact k-means is an NP-hard problem, meaning that doing k-means exactly is as difficult as like finding the passwords of all of your different credit cards. So technically, exact k-means is extremely like computationally demanding. However, there are some approximations that we can make to turn k-means into an intractable problem into a very simple linear scaling problem. And this is essentially boiling down to how do we pick the initial k guesses for my cluster centroids. By far, the most popular approach is just like you pick k random points and and go with it, and you pray that there is a correct solution. However, there are some heuristics which can potentially lead you to better clusterings using this like ON k-means. Those are known as extremely unimaginative name, k-means plus plus. So the idea in k-means plus plus is that basically you will pick a point, now highlighted in green, and then your second guess is going to be a point that is going to be chosen from a probabilistic function that is going to favor picking separated points from that initial guess, which makes sense because your cluster centers should be spread out through the data. And you repeat this process several times until you pick your desired number of clusters. Okay. There are multiple flavors of k-means plus plus. There is like a vanilla version, which is the one that I just explained. There is a greedy version that essentially does like a local optimization around like each green point. And there is like a similar variant, like a more like brute force plus plus that is implemented in the CVP trash package. Now, our approach for k-means is the following. So we are going to be using the same tricks that I showed you before. First of all, we are going to use complementary similarity to identify high and low density regions in the data. And then we are going to use diversity selection to find separated points, separated centroids. So first step, we calculate the complementary similarity. We identify potentially relevant regions in the whole data. We are only going to focus on those. Then we find the midpoint of data, like the central point of your data. That is your starting point. And then after that, you just do diversity selection until you find all of your all relevant candidate centroids. Okay? So at this point, I show you 4 different versions of k-means. Now we come to the problem of how do we validate, how do we show which k-means is better than the other. For that, we're going to use different quality metrics to validate the clustering. We can use the Kalinski-Harabas or the pseudo F metric that essentially quantifies how compact are going to be the final clusters. We can use the silhouette score that essentially quantifies how separate are the clusters. And like other different metrics that essentially all of them evaluate either how compact or how separated are going to be your sets. Basically, what we want is also another answer, like a critical problem, like how do I find the optimum number of data, the optimum number of clusters in my data. So once again, if we can leverage these indices, not only to separate one method from the other, but also to tell me something about the inner structure of my data points, I will be getting like actually something valuable. So essentially, we want bigger CHI and bigger silhouette values, and we want lower MSD and Davis-Golding's values. So how do these behave over different systems? For like 3 model peptides, what we can see with CH, once again, bigger means better, is that a CVP trash is not doing great, but essentially our K-means is essentially the same quality as the other K-means plus flavors. When we see the silhouette score, once again, bigger is better. Once again, CVP trash not doing great, and our method is like essentially competitive, if not better, as you can see for the beta heptapeptide and the billing headpiece on the right. Now, at this point, we really cannot say anything about the optimum number of clusters in the data because all of these indices are essentially monotonically increasing with decreasing K. So all of them will favor essentially one cluster in your set. So they will allow you to compare different methods, but they don't tell you anything about how to identify the optimum value for K. When we look at the MSD, more of the same. Once again, CVP trash clearly not doing great, and our methods essentially, once again, like if not outperforming same quality as the K-means plus. Only when we look at the Davis-Golding index or the DVI, we see some non-trivially monotonic behavior. So this is actually the index that we are going to be focusing on to identify what is the optimum value for K in your set. How do we do that? Well, we propose essentially two different statistics. Now I'm going to be focusing only on this system, on the billing headpiece. So you can either look at the lower, like the absolute lowest value for DVI, and that will give you an idea for the best value for K, or you can look at the local variation of the DVI values, actually the second derivative of the DVI values. So if you find like this, like local minima with like a steepest slopes leading to it, that identifies a K value that is more stable than the neighboring like clustering results. Now, what I want you to focus is that in the green, orange, and blue plots, there are error bars. Those error bars means that we ran CVP Trash and the vanilla and greedy K-mean plus clusterings, and we got different results every time that we run them. And you don't see error bars on the red plot, which is our method, because our method is 100% deterministic. So how big can be these variations? That can be big enough that when you try to identify the optimal number of clusters in your data, you can get wildly different results. So if you look at the greedy, vanilla, and CVP Trash, so for CVP Trash, you can get that in one run, you get 5 clusters, in the other you get 30. So once again, like wildly different results. However, if you do nanny, no matter how many times, you will always get exactly the same solution. So once again, nanny is not only fast and memory efficient, once again, because it is essentially K-means. To me, the biggest selling point is that it is robust and fully 100% deterministic without sacrificing any of the quality of the K-mean plus variant. So if you plan to do K-means, please give nanny a try. Okay? So now, moving to the end of the presentation, I showed you before that the new method that we have to do comparisons for small molecules, which we call icing. So a bit of history in the group. First, when we came out with the method, I wanted to call that a complementary similarity index. So the acronym was CSI. But my students were too young to know what CSI stands for. So they say, let's call it icing because it sounds nice. And I only agree to use icing because now whenever we have a cool result to show, I can tell you that we have, just like some comparison for like a billing and the NOC2, which essentially is like showing that we can reproduce the hierarchical results at K-means quality. So once again, like I only agree to use the icing name because whenever we have something like amazing 2%, I can say that that is just the icing on the cake. My students absolutely hate this slide, but I'm going to be using it forever because I know just like made them like cringe so much. So the biggest application or the most like spectacular application of the clustering for small molecules is essentially finding a way to solve the problem of clustering for small molecules in linear time and memory. So traditionally, if you are doing clustering in drug design, there is a huge chance that you are using Taylor-Butina or sphere exclusion clustering. And the methodology for this is incredibly simple. You have n molecules, which you need to essentially double, and you need to calculate the similarity between every single pair of molecules. Essentially, you are going to fill this matrix with similarity values. You need to define a similarity threshold, essentially how far away do you are allowed to be to define two things as similar. You find the molecule that has the biggest number of neighbors up to that threshold. You include every single neighbor of that molecule in one cluster. You separate that from your data, and then you repeat this until you run out of molecules or things to add to a new cluster. This is obviously, by construction, O(n) squared in time and in memory. And I'm going to be emphasizing the aspect of memory in a second. So if you want to do this in practice, you will probably use RDKit, which is like the free open source, like cheminformatics, like drug design tool. If you want to cluster short of 500,000 molecules, this will take you essentially 10 hours to do, which doesn't seem like a lot until you realize that clustering 500,000 molecules took more than 4 terabytes of memory to build this matrix. So we didn't go beyond 500,000 because of the time, we didn't go beyond 500,000 because of the memory. Like we didn't have enough space in our cluster to allocate more than 4 terabytes to this problem. On the other hand, if you use our method, this is the time dependency. So now you can do 1 million molecules in your laptop in essentially 3 minutes. So this we recently just like released the code and the prepping for this like new clustering method, which we call Bedburch. Once again, as you can see with time dependency, is a truly ON algorithm that also save you a lot of memory while doing so. So what is the trick? Like what is like the secret behind Bedburch? So essentially, we are going to be building a tree. So you start passing your molecules to this tree, and you start allocating them to different clusters. So if a new molecule comes and doesn't fit in a cluster, you just create a separate box for that molecule. Now, when you hit a limit of clusters in one of the three hierarchies, you will essentially build a representative for those clusters, and you start growing your tree from the leaves to the root being the initial point. So at any given point, you're not going to have more than a preset number of boxes at every single level. So once again, this incredibly reduces the number of comparisons that you need to make to do the clustering and allows you to save a lot of memory because we are also not storing all these vectors. We are storing the sum of the vectors because that is what we need to calculate the Ising values. So we're storing essentially the combination of all of these vectors. We find a cluster representative using that like simple formula with a floor function at the end. And essentially the tree structure is essentially guaranteeing that this is an ON algorithm in time. And the trick of adding everything is guaranteeing that you have an ON algorithm in memory. So this wasn't possible before. So this algorithm wasn't possible before because there was no Ising before. And essentially we need Ising to calculate the different properties of the cluster. So for instance, if you want to calculate the diameter of a cluster, this is essentially just calculating the average similarity between every single pair of molecules. Once again, for this before, you had to store everything and do the ON square calculation. Now with Ising, this is just calculating the Ising of the set, and that's it. It's as simple as that. Another important property, calculating the radius of the cluster. Once again, it's a slightly more complicated formula, but now it's just calculating the average separation to the center of the set. And once again, with Ising, we can like trivially do that, just like with that line of information. So we don't need to store all the structures to get this information. I'm presenting this for small molecules, but this can also trivially be extended to essentially to storing and sorting to very large data sets of protein conformations. So once again, also potentially important for the audience. Now, how do we compare Bedburch with the Taylor-Bottina algorithm? First of all, if you see a distribution in number of clusters, changing this like a similarity threshold parameter, we're essentially following the same trend. So both algorithms are finding the same global structure in your data. At the local level, this is comparing the representative from the most populated clusters. We are also identifying, at least for the most populated clusters, the more important molecules that Taylor-Bottina is finding you, but just like a drastically reduced price in memory and time. We can run the same analysis, like the same clustering quality metrics. So once again, for Kalinsky-Harabas, bigger means better. For Davis-Balding, lower means better. And for the Don index, bigger means better. Our method is outperforming, if not of the same quality, over all the important range of similarity thresholds as Taylor-Bottina. We can also check how the position of clusters in chemical space is going to be changing with different parameters. It's just essentially clustering Chembol at different levels of resolution. You can see that we're essentially like able to pinpoint the more like denser regions independently of that like user-defined parameter. So now for the final application, let's say that you have let's say a non-cooperative supervisor in your group. And let's say that your supervisor asks you to cluster 1 billion molecules for today. So when you should have started your PhD to have 1 billion molecules cluster by today? Well, if you're using RDKit First of all, the scale is in years. So should you start your PhD by the time that Gutenberg printed the first Bible? Slightly bigger than that. The fall of the Roman Empire? Not enough. Democracy in ancient Greece? Not quite. I mean, like PhDs are long, but it's by the time of like Egypt, like Egyptians like building the pyramids, it's like not even that. If you want to cluster 1 billion molecules using RDKit, you will need more than 6,000 years. So once again, like PhDs are usually long, but a stretch. Okay? Now, if you want to cluster 1 billion molecules using our method, and I mean, like, I'm getting older, but not 6,000 years old, so we have to do something different. First of all, the scale now is hours. So will it take you longer than the 24-hour Le Mans race? Will it take you longer than like essentially like a full performance day at Coachella? I know, just connecting with Gen Z. Will it take you more than like an actual day of work? So we did cluster 1 billion molecules, and as far as I know, like we're the first group reporting the clustering of 1 billion molecules, and it took 4.3 hours, which is essentially the same amount of time that you need to spend watching the standard version of the Return of the King. So if you start watching the movie, get yourself a snack. Amazing cinematic experience, like 11 Oscars for a reason. Also, you get to essentially finish your PhD in essentially under 5 hours. Okay. We're still working on improving this. We want to get to like Fellowship of the Ring of 2 towers, like level of time. So we want to go like below this essentially. So we are working like new strategies to improve in web but already like the current Python implementation allows you to like go to like sizes of chemical space that were like totally unprecedented until now. So with that, I hope that I convince you that we can compare molecules fast. And if you have a lot of molecules, we can calculate that like even faster. We can do sampling, we can find representative elements, we can apply this once again to chemical space, to protein and DNA conformations. Please give MDance a try if you are interested in dissecting ensembles of conformations for biomolecules. If you have any doubts, complaints, let me know. Remember to like, comment, hit the notification bell, and subscribe if you have any comment or doubts. And finally, with WebVerge, you have the ability now to truly cluster or analyze billion-size level libraries like as a routine task, just like taking a couple of hours with not a lot of memory. Okay? Once again, it is a truly online algorithm in the sense that you can even accommodate, and we have the functionality to accommodate WebVerge to handle infinitely many molecules. So if you are actively generating a set, let it be of small molecules or protein structures, you can use WebVerge to save a lot of time and memory. With that, thanks to my amazing group, which were kind enough to post doing like the One Piece stuff with me, and the people that gave us money for the research. And thanks to you.
All right, we have time for a couple of questions from the audience and from the no.
Thanks, Ramon. That was a great talk.
Thank you.
You said you had an offhand comment somewhere in between where you said that you didn't like RMSD as a measure of similarity.
No, yes.
Can you tell us what we should be using?
This. So I mean,
but in the core of this was MSD.
Yeah.
Is it just the algorithmic cost of doing the square root? That's all.
Yes.
Okay.
Yeah.
Nothing,
I mean, like that ring knows that whenever I go to the Amber meeting, my last slide is always like, please don't use RMSD. So the thing is that if you are comparing two structures, so you use RMSD, like you gain nothing, you lose nothing for that. Now, if you have more than two, it's like if you simply by removing the square root, you gain so much efficiency wise. But what is the point of doing that? I do want to comment like a slightly more technical comment on that. By removing the square root, you lose the triangle inequality. So RMSD is what a mathematician will call like a proper metric. MSD is not because it doesn't satisfy the triangle inequality. First of all, who cares about the triangle inequality? Second, I mean, in the context of analyzing conformations. Second, we started a collaboration with the Lily and Sean group at Pittsburgh, essentially generalizing this not only to comparing conformations, but to like essentially comparing different simulations and doing like clustering of different simulations, and essentially finding like intrinsic like pathways when you're running like multiple simulations like at once. We tried different ways of doing that that are constrained to satisfy the triangle inequality, and we try MSD. The met the methods that satisfy the triangle inequality were unable to find the experimentally observed like pathways, while MSD not only completed the task like way faster than everything else, but was the only method that actually allows you to find those like intrinsic mechanisms. So it seems that you really don't gain anything by just constraining to satisfy the triangle inequality in this context in particular.
We have time for one more question. Sorry, Greg. I was wondering, so when you're defining that vector of your ones and zeros for scoring similarity between molecules, do you ever have some sort of weighting if you know, for instance, that certain interactions are present between a molecule and a protein, and you care more about one of those ones or one of those zeros more than the others?
In drug design, people don't care.
Okay.
If you're studying interactions, that is an amazing point. Like you should care. It will be trivial to incorporate that into a framework.
Was there one more in the back? Do we have time for? Did you have a question in the back? Yeah. Okay,
Thank you.
Thank you for accommodating it. You talked about similarity, removing one protein from the cluster, recalculating similarity. Have you tried that with a cluster of drug protein complexes, where you looked at the similarities of different drugs binding the same protein, and then removing one, see if there's a central conformation that drugs seem to like to target, or the opposite, including the unbound structure and seeing if that's sort of in a separate conformational space?
So we haven't done it with the tools that we have in place. Should be pretty easy to do. It's like the code that we have in place right now will like easily accommodate that, but we haven't done those like experiments yet.
Actually, that's a really good idea
Mhm.
to try. Thanks. Let's thank the speaker one more time for a great talk. And before we all depart, on behalf of Rutgers, we wanted to give you a token of our appreciation
Thank you.
from the PDB. We start out with a calendar.
Okay.
Then we have, let's see, then we have other pamphlets and information about the PDB itself collected here, and lastly,
Thank you.
we have a PDB pen, and this is actually a really nice thermal mug in case I know you don't drink coffee, but you'll find a good use for it. It holds ice cream. Exactly. All right. So thank you very much.